So now that we've learned transcription and the process of transcription, we will go over regulation of transcription. Now, you can, of course, main resource is this lecture, and you can go back to Cooper's chapter 8. Let's talk about regulation of transcription in prokaryotes, and we will start with the lac operon. So what is the lac operon? First, let's introduce metabolism of lactose. Now, so what happens in bacteria is that they utilize lactose. Now, this is lactose right here. Lactose is a disaccharide, meaning that it is made of two sugar units connected to each other, and it gets cleaved by an enzyme known as beta-galactosidase right in the middle, producing two sugar molecules, smaller sugar molecules, or monosaccharides. These monosaccharides are galactose and glucose, and they can be used, mainly glucose, for generation of energy. Okay, so now this lactose right here can also be converted into what is known as allolactose. Okay, so it's a different structure. It is an isomer. Okay, so it looks like lactose, but it's connected differently, okay, by the same enzyme. Okay, now what happens is that this metabolism right here is regulated at the genetic level, or I should say at the gene level. And the process of gene expression of beta-galactosidase was studied and identified by these two French scientists, Jacob and Monod. Okay, so this is, and they discovered what is known as the lac operon. So what is the lac operon? So basically an operon, again, it is a genetic unit. It gets transcribed into one single messenger RNA, and this one single messenger RNA can produce different proteins with different functions from different parts of the messenger RNA. So the proteins that are produced from this messenger RNA, which is produced by the lac operon, are involved in the metabolism of lactose. So one region of the messenger RNA can produce the beta-galactosidase enzyme that cleaves lactose into these two monosaccharides, galactose and glucose, and it can convert lactose into allolactose. Then you have permease. Permease is a protein that is involved in the transport of lactose from outside of bacterial cells to inside. And then you have a third enzyme known as transacetylase, and the particular specific function of transacetylase is not really known so far, but the thought is that it converts some toxic molecules that look like lactose into something that is less toxic or not toxic at all. So anyhow, so what happens here is that you have RNA polymerase binding to the promoter region, and they have transcription of this single messenger RNA producing these proteins. Okay. All right. So this is the lac operon. Again, you have a promoter region, and then transcription of one single messenger RNA producing these three proteins. But there's also another gene that is involved in regulating transcription of the lac operon, and it is a gene that is located at a distance from the lac operon itself, and it has its own promoter region as well. And this gene is known as the LAC-I, and I stands for inhibitor. Now notice that the operon is polycistronic, whereas the LAC-I is monocistronic. Why is it monocistronic? Because you have production of one messenger RNA producing just one single protein, okay, which is known as the lac repressor. So what does this repressor do exactly? Well, it is produced from the LAC-I gene, and what it does is that it binds to a region that is between the promoter and the transcription start site. Okay, it binds to this region and it is known as the operator. Okay, so you have this lag repressor right here binding to the operator, and what does it do exactly? It prevents the RNA polymerase from binding to the promoter region. So what it does is that it blocks RNA, it blocks transcription from taking place. Okay, so how does transcription take place? Well, it takes place by preventing the repressor from binding to the operator. Now, once this happens, the RNA polymerase can bind to the promoter and you can have transcription. And how is the repressor prevented from binding to the operator? By binding to allolactose. So if there is lactose, it gets converted to allolactose. Allolactose binds to the repressor and the repressor is released from the operator. So the RNA polymerase can bind to the promoter, producing the beta-galactosidase and the other proteins. The beta-galactosidase is the enzyme that metabolizes lactose, and bacteria can utilize lactose as well as the products, okay, glucose and galactose. So here's what happens basically. Again, the LAC-I produces the repressor. The repressor can bind to the operator, but if there is allolactose, allolactose binds to the repressor and the repressor that cannot bind to the operator, and you have transcription going on. So this is known as the positive regulation because it is something induces regulation. So this is known as positive regulation. Okay, so you have transcription going on, so that's something positive. Okay, so wait a second. So we need allolactose to make beta-galactosidase, but at the same time we need beta-galactosidase to make allolactose. So which one does come first? So it's like this riddle right here, right? So the chicken and egg, which one does come first? Here's a concept that you need to know in molecular biology as well as in biochemistry. Some promoters are leaky. Okay, so it's like a faucet. Even though it's closed, tightly closed, you can still have some water dripping down. So why? Because the interactions between molecules is really non-covalent, and non-covalent interactions are reversible. So yes, they can interact with each other, but at the same time they can be released from each other. So even though there is no lactose or allolactose, you can have the repressor or you can have the operator free of the repressor, because the interaction between both is really reversible. So yes, in the absence of allolactose, most of the repressor is bound to the operator, but it can be released, allowing the RNA polymerase to bind to the promoter, initiating transcription, producing little beta-galactosidase. Okay, so here's another concept that you also need to know, and that is you have regulatory sequences on DNA. These sequences can regulate transcription. Now these are known as elements, by the way. Okay, so these sequences are known as elements. So whenever you hear the word element, it means it's a specific DNA sequence, and these are known as cis-acting elements. What does that mean? What cis means at the same level. Okay, so basically these sequences can regulate transcription of genes that are located on the same DNA as these elements. Okay, so at the same level of these elements, and that's why these are known as cis-acting elements. Now, examples of cis-acting elements include the promoter itself, the operator as well. On the other hand, now proteins are known as transacting factors. So whenever you hear the word factors, it means we're talking about a molecule like protein. And what does trans mean? Trans means at a different level. Okay, so these trans-acting factors are proteins or molecules that can bind to cis-acting elements on the same chromosome that produces these factors, or a different chromosome as well. So if you have, let's say you have two chromosomes, now the elements, the cis-acting elements would function on the same level of the chromosome. So you cannot have a promoter on one chromosome acting on a gene located on another chromosome. But you can have a transacting factor that can bind to this chromosome or the other chromosome. And these are produced from genes. So these genes are also known as transacting elements. Okay, so the genes themselves, the protein coding genes, are transacting elements, because wherever you put this element on this chromosome or the other chromosome, they can still be functional because they can produce a protein, and this protein can bind to the cis-acting elements on this chromosome or the other chromosome. Okay, so here's another concept that you need to know. Now, so can you mention examples of cis-acting elements that exist on, let's say, the human genome? Okay, think about other examples. Now also think of examples of trans-acting elements as well. Okay, that is, let's say, genes. Okay, so keep that in mind. Now, something else that you need to know is the effect of mutations. Now, some mutations can cause constitutive expression. Other mutations can be non-inducible. So what do we mean by that? Constitutive means that the gene is always on, no matter what, whether there is lactose or there is no lactose, the lac operon is always working. It's always being transcribed. Okay, but there are other mutations that can make the lac operon non-inducible. Okay, meaning that even though there is lactose, there is no transcription whatsoever. Okay, no matter what. So let's talk about some mutations that can cause constitutive expression. I'll give an example and you can think of other examples. So let's say you have a mutation in the LAC-I gene, okay? A change in the nucleotide sequence, let's say, that produces a non-functional LAC repressor protein. This mutation, or this change in the LAC repressor protein would make the protein unable to bind to the operator. Okay, and even if there is lactose, and lactose is bound, even if there is no lactose, I'm sorry, the LAC repressor cannot bind to the operator. It means that the RNA polymerase can bind to the promoter and it can induce transcription all the time. Okay, so this is a constitutive mutation, or a mutation that causes constitutive expression. So think of other mutations that can result in constitutive mutations. I'll give you another example. Let's say that you have mutation in the operator itself. Okay, so the LAC repressor is functional, it's fine, it looks good, but it cannot bind to the operator. Okay, meaning that the RNA polymerase can still induce, can initiate transcription all the time from the LAC operon. So think of other mutations in the promoter region of the LAC operon, promoter region of the LAC repressor, mutations in the galactosidase gene that produces a hyperactive enzyme or a dysfunctional enzyme. Now let's talk about mutations that cause non-inducible expression. That is, even in the presence of lactose, the lac operon is not functional anymore. So I'll give you an example. Let's say you have mutations in the promoter region. Okay, so the operator is free, there is no lac repressor, but the RNA polymerase cannot bind to the promoter, meaning that it cannot really transcribe the gene. Okay, so this is a non-inducible mutation. There's another non-inducible mutation. So you have the mutation in the lac repressor gene itself, producing a protein that is constantly bound to the operator, even if there is lactose. And even if lactose is bound to the repressor, it doesn't release the repressor from the operon, or from the operator, I'm sorry, so that there will be no transcription whatsoever. So this is really how we understand the function of elements or the function of genes. What we do is that we create mutations in these genes, and we see what happens, the consequences. What does happen if we do this? What does happen if we do that? Okay, and this is how we understand how things work. So let's talk about another level of regulation. So first, we talked about positive regulation. Okay, that is, the presence of something would induce transcription. That is, lactose induces the transcription of the lac operon. So this is positive regulation. We'll talk about another level of regulation by a small molecule known as cyclic AMP. Okay, now this cyclic AMP binds to a protein known as catabolite activator protein. What this protein does is that it binds to a region, to an element that is upstream of the promoter. Okay, and it interacts with the RNA polymerase, inducing it, activating it, pushing it forward so that it induces transcription really fast and really efficient as well, producing a lot of beta-galactosidase and all of the other proteins. So this here, again, that's another level of regulation. So now the thing is that in the presence of CAP, and if CAP is bound to is not bound to cyclic AMP, now this CAP cannot bind to that element upstream of the promoter region. Okay, so there would be some transcription. Okay, that is in case there is lactose. But if there is lactose and there is high level of cyclic AMP, then CAP can bind to cyclic AMP can bind to CAP. CAP binds to the to that element, inducing a lot of transcription of the lac operon. Okay, good. So again, if there is cyclic AMP binds to CAP. CAP activates the RNA polymerase even further. So how is cyclic AMP produced? It's produced enzymatically by an enzyme known as adenylyl cyclase, and this enzyme, adenylyl cyclase, converts ATP to cyclic AMP. Okay, so if this enzyme is active, you would have a lot of cyclic AMP. Great. Now the thing is, this enzyme is regulated by glucose negatively. Okay, so in the presence of glucose, glucose would inhibit adenylate cyclase or adenylyl cyclase, disabling it from producing cyclic AMP. This is known as negative regulation. That is, the presence of something negatively regulates or represses transcription. Okay, so lactose inducing positive regulation, because when it's present there is transcription. Now glucose, on the other hand, induces negative regulation, because if it's present there would be no transcription of the lac operon. Okay, so why is it that glucose does that? Because if there is lactose and there's glucose, bacteria would prefer glucose over lactose, and it makes sense, right? Because glucose is there. It is an immediate source of energy. On the other hand, lactose needs to be hydrolyzed so that it can be utilized. So bacteria would say, you know what, I'm going to handle glucose because lactose, I have to hydrolyze it. So it says, no, forget about lactose. Let me consume glucose. Okay, so there is preference that bacteria have. So again, ATP is converted to cyclic AMP by adenylyl cyclase, and this adenylyl cyclase is regulated by how much glucose there is in the cell. So if there is high level of glucose outside of the cell, it goes into the cell, it inhibits adenylyl cyclase, and there would be low level of cyclic AMP. Cyclic AMP cannot bind to CAP. CAP in this case cannot bind to the element that is upstream of the promoter. So there would be some transcription, but it's not really efficient because the RNA polymerase is not really pushed by the CAP protein. On the other hand, if there is low glucose, in this case bacteria would say, I don't have much glucose, let me utilize something else that is lactose. So adenylyl cyclase is active in this case because it's not inhibited by glucose. It produces a lot of cyclic AMP. Cyclic AMP can bind to CAP. CAP binds to DNA, inducing a lot of transcription of the lac operon. So let's talk about different situations here. Let's say there is no lactose and there is glucose. Now in this case, there's no lactose, right? In this case, the lactopressor would bind to the operator, right? And RNA polymerase cannot bind to the promoter region. At the same time, there is glucose. It means that there is inhibition of adenylyl cyclase, low level of cyclic AMP, and CAP is free of cyclic AMP, right? So it cannot bind to that region that is upstream of the promoter region, right? So there would be no mRNA transcription, mainly because the lactopressor is bound to the operator. Now, let's say that there is lactose and there is glucose. Now, preference is for glucose, right? But at the same time, since there is lactose, now this lactose, or I should say allolactose, would bind to the repressor, releasing it from the operator. And the RNA polymerase can bind to the promoter and it can induce transcription. But the level of transcription is really low because CAP is not bound to cyclic AMP. So it cannot bind to that element upstream of the promoter region, and it cannot induce, as a result, the RNA polymerase is not really induced to work very efficiently. Now, but if there is no glucose and there is lactose, the repressor is now free of the operator. It doesn't bind to the operator. At the same time, CAP is bound to cyclic AMP because adenylyl cyclase is not inhibited. There's no glucose. CAP binds to the adenylyl cyclase is active. It produces cyclic AMP. Cyclic AMP binds to CAP. CAP can bind to the DNA upstream of the promoter region, and it can induce the RNA polymerase to transcribe the lac operon, producing a lot of messenger RNA, and transcription is really very high, very efficient. So what I want you to know now is that if you understand how the lac operon works, and it is really a simple genetic system, you would understand how transcription function or how it works, and regulation of transcription. So the idea here is that gene expression is regulated by regulatory proteins. And these proteins can do the following. They can guide the RNA polymerase to the promoter region. Okay? They can also guide and recruit other regulatory proteins to the promoter region. Now, these regulatory proteins can also strengthen or stabilize the interaction between the RNA polymerase as well as other proteins to the promoter region. Okay, these regulatory proteins can activate the RNA polymerase. Okay, They can also create the open promoter complex, facilitating transcription. Or these regulatory proteins, by the way, can do the opposite. So they can prevent the RNA polymerase from binding to the promoter region or other regulatory proteins. They can weaken this interaction or prevent it altogether. They can inhibit the RNA polymerase from moving forward and initiating transcription. They can create the closed promoter complex, okay, or they can prevent the proteins that create the open promoter complex from working. So that's how repressors function. So all of these effects are really modulated by regulating these non-covalent interactions between proteins, whatever these proteins are, and DNA. Okay. Now, again, it's all non-covalent. So you have these proteins, regulatory proteins, that can bind specifically to the major groove of DNA, and these interactions between DNA and proteins can modulate and affect these non-covalent interactions between the two.